Hello everybody. Today I will focus your attention on biosynthesis of amino acids and other biomolecules that are derived from amino acids. Next time I will talk about the degradation and biosynthesis of nucleotides. Now, to start the topic of amino acid biosynthesis, I guess it is good to realize what are the main components that would be used for synthesizing amino acids. And if we go in a way backwards in principle of amino acid degradation, we will need a nitrogen or amino groups or some sorts of nitrogen. And we're also going to need a source for the carbon skeleton of the amino acids. So what are the sources of nitrogen? Where would the biosynthetic enzymes get nitrogen from? Now, the main source of nitrogen is actually glutamate. When we aminate alpha-ketoglutarate, glutamate is generated. And glutamate is the main resource or source of nitrogen for amino acid synthesis. Glutamine can also contribute to that, but the contribution is really small. In bacteria and bacteria and other protozoa, they also utilize different processes that are not available to higher organism, and that is, they facilitate nitrogen, so-called nitrogen fixation from the atmosphere, which is an amazing process that plays a major role in the cycle of nitrogen and oxygen, for that matter, in the cycle of life on our planet. And because of that, and because of the fixation of nitrogen from the atmosphere, and then the involvement of that nitrogen into biomolecules in bacteria, and then from there to plants, and then from plants to animals, and from plants and animals to our bodies, basically nitrogen fixation has a major role for amino acid synthesis in our bodies as well, but indirectly, because obviously we cannot use atmospheric nitrogen and fix it and use it for any of our purposes. Now, in terms of carbon, what are the sources of carbon? We could look back to the figure that describes the degradation of amino acids, and we could speculate that any of these metabolites could be used potentially to generate amino acids. For some of them, that is true. For others, though, that will not work. So, but I will focus on this one a little bit later. What I wanted to say, though, first is that the de novo synthesis of amino acids is not as prevalent in the cell as, for example, the synthesis of fats or carbohydrates. And the reason for that is because the major source of proteins, of amino acids used for protein synthesis, is actually recycled amino acids. Proteins in our body are constantly synthesized and broken down or degraded. When proteins are degraded, the resulting amino acids are not degraded further down to carbon dioxide and water, but they are recycled. So if a protein is degraded to its amino acids, these amino acids are immediately utilized by the ribosomes to generate new proteins. So if you remember when I talked about amino acid degradation, I only said that amino acids are used only for about 10% of our energy needs. In other words, it depends on the diet of a person, but in other words, proteins are rarely used for energy. Amino acids are rarely degraded to carbon dioxide and water. And that is because they have a different purpose. They are used to build proteins, and protein synthesis and degradation happens constantly in our body. And recycling of amino acids is a very common occurrence. Also, if you think about it, when you consume proteins with your diet, they will be degraded to amino acids. And if these amino acids, and they will not in normal organisms, under normal circumstances, for the most part, these amino acids will be immediately utilized for protein synthesis. These amino acids will not be degraded to carbon dioxide and water, but they will immediately be used for the synthesis of our own proteins. Just because you have eaten a plant protein, they're still made by the same amino acids that our proteins are made. Even though a plant protein or an animal protein doesn't match our protein exactly, that doesn't matter. They will be degraded to the amino acids, and the amino acids are the building blocks that will be used for building our own proteins. So amino acid recycling is very prevalent in the cell, and that is good also because 10 of the 20 amino acids cannot be synthesized by our cells. They can be synthesized in plants and other organisms, not higher organisms, but we cannot make them, and we have to consume them with food. And these amino acids are valine, leucine, isoleucine, lysine, histidine, tryptophan, phenylalanine, tyrosine, methionine, and threonine. Now, there are certain ways to try to remember these. I am not going to attempt to tell you how do you memorize these, but there are certain ways to memorize them, though. I do want you to know them, though. For me, it is important that you, as biochemists, that you know the 10 essential amino acids, just like to know the essential fatty acids. Okay, so 10 of these we cannot make. We have to take them with food. All right, so let's transition now to the place where we're going to talk about the de novo synthesis. And like I said, this is not very common. It doesn't mean that it doesn't happen. It does happen. But this is not the major way of getting amino acids. The body would much rather consume proteins from plants or animals than to synthesize de novo amino acids from its precursors. That would mean that you will pull away metabolites from glycolysis, from TCA cycle, and the pentose phosphate pathway, which obviously have importance for energy production and for other processes in the body. So what are the metabolic precursors for amino acid synthesis? These precursors can be united in 3 major groups based on the pathways they're found. So we have 2 precursors that come from the TCA cycle, and those are oxaloacetate and alpha-ketoglutarate. Oxaloacetate, by amination, when it's aminated, becomes aspartate. When we aminate alpha-ketoglutarate, becomes glutamate, and then from there we make other amino acids. From glycolysis, the precursors are pyruvate, phosphoenolpyruvate, and 3-phosphoglycerate. Now, I have no intentions to make you memorize exactly which amino acids comes from what, but it will be helpful if you know, at least in general, what are the amino acids. Somebody might say, well, that's no help. You're telling me it will be helpful, but do I now memorize them or I don't memorize them? Well, let me put it this way. I don't know them. I don't know which amino acids. I don't remember which amino acid comes from which precursor. But I know some. For example, I know that oxaloacetate is made out of, aspartate is made out of oxaloacetate, glutamate from alpha-ketoglutarate, alanine from pyruvate. These are key amino acids, and I know that they come from these, that these are junctions used in these pathways, and you can convert alanine to pyruvate and contribute to glucose production. And alpha-ketoglutarate and oxaloacetate are important metabolites not only for the TCA cycle, but you've seen them being involved in amino acid degradation, in urea cycle. So these are important amino acids to know where they come from. But whether I know proline, that to me is not a big deal. So I still want. That's why I'm advising you to kind of read through them, just try to connect them, but don't waste hours now to try to memorize absolutely every single amino acid where it's coming from. That's not my intention. Okay, so 3-phosphoglycerate is the other, and you can see serine and glycine and cysteine, and pentose phosphate pathway is the third pathway that contributes a precursor, and that is ribose 5-phosphate, which is used for histidine production. Now, the interesting question, though, is that while we can use these, 1, 2, 3, 4, 5, 6 precursors to make all 20s, now, but we said also that mammals cannot make all 20s. They can only make half of them. So, in real life, which precursors do mammals use for amino acid synthesis? So in order to answer this question, you should look at this table, and where you realize that alpha-ketoglutarate is actually used for amino acids that are non-essential for the body, such as glutamate, glutamine, proline, and arginine. So we can synthesize these from alpha-ketoglutarate. So alpha-ketoglutarate is a precursor mammals can use. 3-phosphoglycerate, the same for 3-phosphoglycerate. We can use that one to make amino acids. Oxaloacetate. Oxaloacetate is used to synthesize 5, but we can only make 2 of those. So still, we can use oxaloacetate. Pyruvate in general can be used to synthesize 4 amino acids, but only 1 is a non-essential, meaning only 1 can be generated in our own bodies, and that's alanine. Phosphoenol pyruvate and ribose 5-phosphate can be used for, they are used for amino acid synthesis, but not in mammals. In other organisms, of course, but we cannot use them. So, in essence, the only precursors in our bodies that can be used for amino acid synthesis are alpha-ketoglutarate, oxaloacetate from the TCA cycle, and from glycolysis, 3-phosphoglycerate, and pyruvate. So here, I synthesize basically the entire chapter into this slide. Instead of looking at the individual pathways of amino acid synthesis, I basically concentrated your attention on this slide, at least for you to know what are the precursors in mammals. And these are oxaloacetate and alpha-ketoglutarate, pyruvate, and 3-phosphoglycerate. These are the precursors for amino acids in mammals. All right, now I'm going to transition now into the synthesis, or giving examples of some bioactive molecules that are synthesized from amino acids. Now, this example here shows how glycine is used for the synthesis of porphyrins and ultimately the heme group. And as you know, heme group is a very important component of hemoglobin and cytochromes. Hemoglobin is important in the transport of oxygen to the tissues, and cytochromes is important for the transfer of electrons from one electron carrier to another electron carrier. So they both have a very important role. Hemoglobin, though, is recycled. This is not a structure that once it's made, actually nothing pretty much in the body, once it's made, it stays this way. Everything pretty much changes, and molecules that are once synthesized, they usually have a certain half-life, and they, after a while, they get sometimes quick, sometimes very slow, but they get degraded and regenerated. And heme is no different. The heme is no different. Heme is degraded to a compound known as biliverdin, and biliverdin is then converted to bilirubin, and bilirubin is a metabolite that is often looked at when you do blood studies or urine studies, because it tells you about the liver function, and it's usually, that's what it's usually used. If bilirubin start rising, that means that there is an issue in the liver. There is an inflammation in the liver, which means that bilirubin, more of the bilirubin now transitions into the blood. The skin turns yellow. The eyes, the sclera of the eyes, the white part of the eyes turns yellow. The urine turns brown, dark yellow brown color, and that is a sign usually of a liver damage of some form or liver inflammation. If there is an obstruction somewhere in the biliary duct that does not allow for the bile to enter the intestines, then the intestines actually become almost colorless. I mean, they are not exactly colorless, but they become this pale gray color. And that's a sign, again, that there is a problem in the processing of bilirubin. And it points towards the liver or the gallbladder or the pancreas. It's just based on what the clinic is, I guess. But I don't want to go into too much detail there. It's important for you to know that this is a dye, and this is a dye that basically stains the urine yellow, and the stools are stained brown as a result of this dye. And this dye is produced from the heme group when erythrocytes and other enzymes that contain the heme are being degraded and repurposed or processed or regenerated. So that's the connection I wanted you to see, from glycine to the heme group, from heme group to the dye, and that dye then goes into the stools or into the urine and gives that typical color that we are all familiar with. Other examples of bioactive molecules that are derived from amino acids are the hormones and neurotransmitters that are made out of tyrosine. Remember, tyrosine is a conditional amino acid, conditionally essential amino acid. It's made out of phenylalanine, and so if we don't have phenylalanine, then we cannot make tyrosine. If we have phenylalanine, we can make tyrosine. And then tyrosine is used to generate dopamine, norepinephrine, and epinephrine. These are all neurotransmitters, and some of them, such as the epinephrine, norepinephrine, can also act as hormones. Another example is the gamma aminobutyric acid, or GABA, which is another neurotransmitter that is generated from glutamate. Histamine, which plays major role in the allergic reaction, in the allergic response, is made out of histidine. Histamine is the hormone that, when we have an allergic reaction, is released in the affected area, and what happens is that it opens up the capillaries a little more, and then liquid and proteins and cells transition through the capillary walls into the tissue that leads to swelling, stretching of the skin, which causes the itching as well. More blood comes to the area to bring more cells, and that causes the area to become reddish. So the itchiness to a point of even pain sometimes. The readiness, and of course the swelling also can affect the function of that area, or formation of little bumps that are so typical for local allergic responses. So that hormone, histamine, is made out of histidine by decarboxylation, as you can see here. Another one is serotonin. This is the, I call it the happy hormone because when it's a neurotransmitter, it's not a hormone, I'm sorry. It's a neurotransmitter that if it's in deficit, people usually are depressed, and so that's why a major group of all antidepressants aim at increasing the amount of serotonin in the synaptic cleft, so that the feelings of happiness and satisfaction are increased. This is a hormone that is usually released with after you eat, and that's why when you eat you feel good and you like to relax. Some people falsely accuse turkeys, telling that they're rich on tryptophan, and because they're rich on tryptophan, that means that you're going to make a lot of serotonin. That's I believe well, I want to say I don't believe to this at all. I don't believe this at all. I mean, first of all, you can, anything that you eat in such quantities during a Thanksgiving celebration would make you sleepy, or it would make you relax or feeling good. So to blame the turkeys for the high concentration of tryptophan in their meat, and because of that high concentration of serotonin, that seems silly to me. Anyway, let's move on. Arginine is used to generate citrulline, which we know how citrulline is used, but more importantly, nitrogen oxide. And nitrogen oxide is involved in blood clotting and the regulation of blood pressure. So it's a very important compound. And it's not even a larger molecule. It's nitric oxide. It's a tiny molecule, but has a great role in the normal function of the heart rhythm and blood pressure and the activity of the blood vessels. All right, so at the end, I just wanted to finish this discussion with some general principles of the amino acid biosynthesis regulation. That regulation is usually allosteric. That means that we're going to involve a separate site other than the active site that will bind usually a product of some form, downstream product, which we know as feedback regulation. For example, in this case, you have one of the downstream products of threonine is isoleucine, and the synthesis of isoleucine inhibits its own synthesis from threonine. So if you have too much isoleucine, that will inhibit the synthesis of isoleucine from threonine. That's an example of a feedback regulation, a simple feedback regulation. In addition to that, though, we observe cases where we have multiple products, as you see here, that inhibit the same enzyme. This type of regulation is still considered feedback regulation, but it's also known as concerted regulation, where multiple products inhibit the same enzyme. Another level of regulation that makes things more complex is enzyme multiplicity. And in that case, we basically have one enzyme that has multiple isoforms. They all catalyze the same reaction with the same product, but they're inhibited by various products that are produced down the reaction or down the pathway. And as you can see here, for example, the enzyme that converts aspartate to aspartyl beta phosphate has 3 different isoforms, and they are all affected by different metabolites downstream produced from aspartate. And also various cells have only one, perhaps 2 of those, but the reason why we have multiplicity is so that we can have a different level of regulation. And if you want to turn off only the expression or the use of aspartate in a certain number of cells that have only A3, you can do that through threonine in this case. But the others, in other cells, they will not be affected by that type of regulation. So enzyme multiplicity allows for more complex regulation. And the last type of allosteric regulation is the sequential feedback, which kind of is represented in this figure as well. You can see, for example, threonine, how it affects different levels of the same pathway that synthesizes from aspartate. And in a way, you can think of it as a sequential. You can based on the concentration of threonine, you can affect either this reaction or this reaction or this reaction. And for that reason, we call it sequential regulation. All right, so this was in general terms all I wanted to say about amino acid biosynthesis. Like I said, it's a short presentation this time. Next time I will talk about nucleotide biosynthesis, degradation, and release from the cells and from the body. So until next time, I hope everything is well, and if you have any questions, please let me know. Thank you.